Hello, everyone. We are Team GoldenEye, and our project is polarization-based optical background nulling for SP-Iris biosensing system. Our instrument is designed to detect proteins. So please bear with us while we quickly and efficiently explain how and why our instrument works. Proteins are the machines of the human body. They make the human body work. If the proteins are no longer working, a problem arises in our bodies. And any disease is a result of a protein malfunctioning in our body. For example, Alzheimer's is caused by a misfolding of a protein. Diabetes is caused by a malfunction of insulin, which is a protein. And HIV is caused by a malfunction of antibodies, which are the proteins used by the immune system. This is why we must detect proteins. And we must use instruments that are fast and accurate in order to find solutions as quickly as possible. The challenge, however, is that proteins are very small, approximately 2 nanometers in size, and thus they cannot be resolved with visible light. However, nature has already created a biodetector for us in the form of our immune system. Fortunately for us, these antibodies in the immune system, antibodies are the proteins which bind and indicate any foreign body. Fortunately for us, these antibodies can also bind to things that are detectable in a microscope, namely gold nanoparticles. Gold nanoparticles are perfect for detection because they have a resonance frequency which is in the visible spectrum. Many instruments currently use gold nanolabels, but these nanoparticles are also very small, approximately 20 nanometers in size. Thus, these instruments require high magnification, and it's very difficult to focus on the particles. For this reason, neither the stage nor the data collection process can be automated. It also takes a lot of time and a lot of labor. Our solution to the problem is to use a background nulling technique with circular polarization and gold nanorods. This allows our system to use a lower magnification and allows us to overall automate the system. Final system has an increased throughput without affecting the apparatus precision.
Okay, so I'll be going over how the background nulling works. So you have your gold nanoparticle on a slide, proteins are binding to it, and you shine circularly polarized light on it. So when circularly polarized light hits the silicon dioxide, it's reflected back and it's still circularly polarized. We pass it through a quarter wave plate and then it becomes linearly polarized. If you shine circularly polarized light on a gold nanorod, however, or any rod-shaped object, the reflected light will be predominantly linearly polarized. And when you pass it through the same quarter wave plate, it becomes circularly polarized again. So if you look at this with polarizers, the linearly polarization will be blocked, while the circularly polarization will partially go through. So this means that the light reflected from the silicon dioxide from the background is not passed through, while the only light that does get passed through is from the gold nanorods, which reduces our background which increases our signal to background ratio.
So to implement this technique, we built a setup which incorporates the polarizers and the background nulling technique. We've also developed an image processing software which analyzes the images that are taken and counts the number of nanoparticles on the surface of the substrate. In addition, because of the novelty of the technique, it was important that we incorporated an electromagnetic model in order to ensure that we're detecting the correct particles of interest. Oh, I lied. It's my turn. So my contribution to this project was microscope design and implementation. This involved both designing the Z-stage mount configuration as well as choosing an optimal objective and camera choice in order to decrease our magnification, increase our field of view, and be able to automate the system. We also needed to be able to maintain the particle detection capabilities, so that was important. Once I chose the components and built the instrument, I also needed to align the instrument. These alignment components were both even illumination over the surface of the sample, as well as making sure that the entire image was in focus so that we could detect particles in multiple spots at a time.
So the hardware and hardware control. So there are three main hardware components to the GoldenEye system: the camera, the Z stage, and the controller box. And as well, there's also the LED. So for our hardware, the Z stage, we chose the PI Myco's translation stage, the VT21S, which is motorized by a stepper motor and has a repeatable resolution of 0.05 microns. This is controlled by the Pulse Box NT controller. For our camera, we chose the Point Grey Grasshopper 3. This features a CMOS sensor. And lastly, we use a 660 nanometer LED and LED driver. And all these hardware components are controlled through our customized GoldenEye GUI. The Z stage control consists of 5 functions. The functions are used to initialize the Z stage and send commands to the controller. The camera control consists of 2 applications: to configure the camera, save images, and lastly to open and generate a live preview window. Additionally, the GUI displays data output, user messages, and contains file management and analysis options. And basically, the GUI is where the software and the hardware are integrated together.
So with the help of hardware, we can acquire an image of a chip. There are lots of information inside this image, and that's why we need to use image processing tools, including spot locator, ZIFT algorithm, and EM model to acquire and analyze these informations. Usually, people make arrays of spots on the chip and bind different kind of proteins to different spots. In order to know the concentration of proteins, we first need to use the spot locator algorithm to identify the spot locations. The idea behind this algorithm is to remove everything else in the image as much as possible except for spots in order to perform a precise detection. However, only knowing the spot position is not enough. We also need to know how many particles are inside each spot. In our project, we use the commercial ZIFT algorithm in order to count for spots. In order to count for particles. And this algorithm is inspired by the human vision system, so it is invariant to scaling and rotation. The combined result of the ZIFT algorithm and spot locator are displayed in the image on the right.
So the SIFT algorithm counts the particles, so it returns you a number. However, because of the novelty of the technique, we didn't have any data that told us how the images should look like. So the SIFT algorithm returns us images that look like this on the left. To verify that those come from gold nanoparticles, we implemented the EM model, which simulates the background nulling technique and tells us what they should look like, and this is the result of the simulation. The fact that they're similar tells us that we are indeed looking at gold nanoparticles. So once the image processing and the hardware control were implemented, we put the two together to achieve the main goal of our project, which was to automate the system. To do this, we implemented the Autofocus algorithm, which focuses the system by moving the Z stage into various positions and counting taking an image and counting the particles in the image at each position. So the result is this curve, which shows the number of particles as the function of the position of the Z stage. As you can see, it has a very clear maximum, which corresponds to the focus. The important thing here is that it has this wide range where you still remain in focus. This is known as the depth of focus. And the previous IRIS system had a depth of focus of 300 nanometers, which is very small, which is what prevented it from being automated because the Z stage just doesn't have the required accuracy. Our depth of focus is about 7 microns, which allowed us to automate the system and showed that the background nulling technique actually works. And we have a video. If I can get the mouse. sorry, I'll have to have some technical issues. Fortunately, I assumed I'll have them. Okay. Sorry, I don't see the file. Oh,
You only ask questions.
I just found it.
What was your assessment process like for choosing the keyboard protection? Why did you choose Selim?
We first use several different ways, like 2D Fourier transform, and then we use the covariance matrix. And we found that they are very insensitive with the term of the z-stage height. We found that ZIFT works best, so we choose ZIFT.
How long does this process take?
The-
You mean the autofocus?
Yeah, with all the Z-stage positions.
Like autofocus take like about 15 seconds for each slices. If you want to do that 20 slices, you just multiply that by 15 seconds.
Take the chip and place it on the stage. Adjust the stage so the beam is in the center of the chip. Open MATLAB. Run the GoldenEye GUI. Click Live Preview to see a live image of the sample. The Z-stage can be controlled through the GUI. Adjust the position of the sample by clicking on the Move Up and Move Down buttons until the image looks in focus. Click Autofocus to more precisely focus the image. The autofocus moves the Z-stage in increments and finds the focused position. Finally, click Run Analysis to count the number of nanoparticles. The number of particles is displayed at the top of the image. Congratulations, you have now found all of the protein.
So as we described, we've designed and created a system that is automated by incorporating optical nulling technique and being able to overall automate the system. We think that the system is better for detecting proteins because it overall decreases the time that it takes and decreases the manpower. So that's our system, GoldenEye.
All right. What is the cost of the system and what's the driving factor?
Okay. The cost of the system is approximately $7,500. The driving costs are the camera, the polarizers, and the Z-stage. Because the Z-stage is automated, the polarizers are very complex components, and the camera is expensive. So, yeah.
Why did you choose that specific camera as compared to other CMOS cameras?
You can talk about the combo of objective.
Oh, okay. So the main thing was that we needed a combination of objective and camera that increased the field of view. There was that image with the different sizes of the field of view. So the camera choice was just mainly the pixel size, which is what goes into determining the field of view.
Yeah, 2048 by 2048 pixel count.
One more. Yeah.
Are those gold beads or gold nanoparticles attached to the protein that way you can count them?
Yeah, so if I go back to the slide, it's more like the proteins are attached to the gold nanorod rather than the other.
It's possible that they are false or not, because not all the gold beads are attached to the protein, so that could be false or not.
So the proteins.
So it's actually much more robust because the gold nanorods are attached to antibodies, which are attached to the protein. So it's a long process to make that sample, but it's much more accurate. I
The
guess.
reason I'm asking this was a few years back I was involved in a research project that was precisely all these floating around that was for detection of several diseases.
Yeah.
And that problem arrived, but I don't know whether that's solved or not. So
Oh.
Okay.
we are not sure that all those particles are.
Yeah, we didn't do a lot of the bio, but
Right. No, it's sort of an open research question, and
Yeah.
sort of the point of building an instrument like this is to figure out whether it works or not. This technique. Yes. Yes.
Thank you.